Well, hello, everybody. Welcome back again to the next chapter in our progression through the semester. Today, we are looking at a large group of organisms that represent probably the most diverse and most complex group of organisms that we will look at this semester. In fact, you can point to them and perhaps say they are the main reason why we have the three-domain system, or one of the main reasons why we have the three-domain system instead of the old five-kingdom system. And this is the protists. The protists are so diverse in form and function and structure and just about everything else that you can imagine. Perhaps the only group of organisms that are more diverse than the protists is the ones we looked at last week, the prokaryotes. And so while we can say that the prokaryotes are pretty much everywhere, we have to say with the protists they're almost everywhere. They are certainly a very, very diverse group of protists. And as a consequence, the taxonomy of protists has been through a number of changes over the course of the time, at least, that I've been teaching this course. And where there are substantial changes to be made in the taxonomy, this is usually where it turns up in trying to deal with the protists. I've tried to incorporate the most recent information as much as I can, and certainly in keeping with what the textbook contains. But I've noticed that in the textbook this time round, they've actually dropped a couple of groups from discussion. They're actually incorporated in a couple of the supergroups. So I've still got them included in here, just so that you're aware of the fact that they exist and they are actually very important parts of the supergroups that are discussed as we go through this chapter. So there's a little bit more information in here than you're going to find in the textbook, and that's because I want to make sure that our look at the protists is as complete as possible, so that in the future, if you should ever encounter a class where you're looking at the protists, you're not sort of blindsided by being introduced to some form of protists that you've never heard of before. As it is, you probably will be anyway, but I like my coverage of the topic to be as complete as I can make it, so it's in there. Anyway, onwards and upwards, let's take a look at these very interesting, sometimes confusing, always diverse organisms. So when you start looking in any water, and if you think back to your biology 1 class, particularly if you had a wet lab, you may have had an opportunity to look at some pond water, and you would have seen a huge diversity of protists in there. Or if you didn't look at pond water, but you did a lab similar to the sort of lab that we do in Bio 1, where you get a chance to look at a whole variety of different types of cells. The largest group of cells that we typically have are the protists, or representatives of that group we used to refer to as protista. Now we just refer to loosely as protists. And that's because there is such a huge diversity of these organisms. And that's really the reason why we don't have a kingdom protista anymore. Remember that under the phyllo code, cladistics, the idea is to come up with a monophyletic clade. And the problem with the old kingdom protista, with photosynthetic, non-photosynthetic, heterotrophic, and then a mixed bag of organisms called mixotrophic, which flip backwards and forwards between photosynthesis and heterotrophism, it was very difficult to imagine what a single common ancestor, this huge diversity of protists, and then you have to layer on the fact that within kingdom protista, you have unicellular, colonial, and multicellular representatives. Just trying to imagine what the single common ancestor that would sit at the base of the tree would be for this huge diversity of organisms was nigh on impossible. And in fact, when we do the genetic analysis that leads to the current phylogeny, it's almost impossible to place a single common ancestor at the base of a tree that would represent the diversity of protists. So the kingdom Protista could not stand under the phylocope. And so we have struggled to try and come up with a system that would satisfactorily classify protists. And we've been through a number of different systems. We right now use a system called the 4 supergroup system. The supergroups are considered to be super kingdom classifications, as best as I can tell from reading the literature. Part of the problem is in creating the 4 supergroups and then placing the recognized kingdoms and so forth underneath that, no one has really decided where in the taxonomic hierarchy each of the clades should reside. Should they be referred to as kingdoms? At one point we had what was known as the 8 kingdom system. And then we had a system that had tentative kingdoms. It's been very difficult to try and get a unified taxonomy of protists, but the 4 supergroup system at least tries to place the protists and other eukaryotes into some clades that we can support using genetic analysis. So as we work our way through this chapter, I'll talk a little bit about the genetic analysis, not a whole lot, because that would be the subject for a whole class, I suspect, in its own right. And we obviously don't have the time for that. But we'll discuss a little bit about the genetic analysis that has led to these supergroups and just what support exists for each of the groups along the way. So here you can see a diversity of protists, everything except this strand and similar strands like it. So that strand, this one over here, these big ones are protists, but these smaller strands, those are actually cyanobacteria, but everything else that you can see in the picture is a protist of some description. There are diatoms, an amoeba, an algae, in fact, several species of algae. There's these spherical algae, the elongated algae. Everything here, except the cyanobacteria colonies, are protists. And so you can see that we're starting to deal with a diversity of photosynthetic and non-photosynthetic protists. And therein lies part of our problem. So I've talked about this already in my introduction. Let me just show you some representatives. Some of these you may have seen. At least I hope you've seen Stentor or something like it. Stentor is a ciliate. It's a cousin of Paramecium and Volvox and, well, not Volvox, sorry, Vorticella, which you may have seen in Bio 1. Those are common ciliates that we have as part of the cell lab. Then we have a dinoflagellate that is photosynthetic. The red color is the presence of a pigment that supports photosynthesis, but it gives it its red color, and there's a reason for that. Then we have a multicellular quote unquote protist. This is a member of the red algae. This one grows at some depth, hence the red color, but you're going to see that there are other representatives of red algae that look almost black or at least dark green because of the significant amount of photosynthetic pigment that they contain. And there are reasons for that as well, more to come. And then of course we have our typical algae. You've probably seen Spirogyra in Bio 1, and then you've probably seen duckweed and other algae floating on the surface of freshwater lakes and elsewhere. These are all protists. And you start to recognize, if we're looking at these 4 pictures, that you've got a huge diversity of organisms in this single kingdom that we referred to as Kingdom Protista, something that created all of this issue with where we placed them. When we start looking at the cellular level, we also find a huge diversity of different types of structures. So we know that there are uniform structures that are recognized within eukaryotic cells. So you see a small list of them down here: the nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes. You should be familiar with all of those. You've encountered them before. But what you find is that some protists have unique organelles that are not found anywhere else. So if you've seen a paramecium, and hopefully you have, under the microscope, you may have seen what are referred to as contractile vacuoles, something that is an organelle uniquely found in ciliates. And these are organelles whose job is to pump water out. It is a way for these types of protists to osmoregulate, to control the flow of water, because they live in a hypotonic solution. The water wants to flow in. They can't stop it, so they collect it and pump it out through these contractile vacuoles. And you find that this is the sort of thing that is not unusual in some groups of protists, that there are small groups of protists that have unique organelles that are not found anywhere else amongst eukaryotic cells. So not only is there a diversity of protists, but even within the protists, whether we're talking about unicellular or multicellular or colonial protists, some protists even have some features that are unique to themselves and therefore separate them out from other protists. And we'll see some of those things along the way as well. So we have to account for all of that as we're trying to establish a phylogeny for protists. And you can see that this is becoming pretty complicated. And then I've already mentioned the idea of photosynthetic and non-photosynthetic protists and mixotrophic protists. So let's just nail that down in terms of their nutritional diversity. So photosynthetic protists are photoautotrophs. They possess chloroplasts and they carry out photosynthesis. They may not necessarily be green. As you'll see, there are brown photosynthetic algae, there are yellow photosynthetic algae, there are red photosynthetic algae, and there are green photosynthetic algae. All of them contain chlorophyll, both chlorophyll A and B as the photosynthetic pigments, but they also contain accessory pigments or auxiliary pigments that are of different colors because of where they carry out photosynthesis, and that changes their color as well. So again, we're now starting to look at some complexity even amongst a single group of protists. Then we have the heterotrophs that go about capturing other organisms and using them as food resources. And again, we see a wide range of behaviors even amongst the heterotrophs. More of that to come. And then we have this rather unusual group of protists that are described as mixotrophic. They do have photosynthetic apparatus. They have chloroplasts. They are perfectly capable of photosynthesis, but they can also capture and eat organisms as heterotrophs. And sometimes they'll do both behaviors at the same time. Sometimes it depends on the light regime. If there's enough light, they're photosynthetic. If there's not enough light, they flip over to heterotrophism. And they'll flip backwards and forwards depending on the light regime. So again, huge diversity at the nutritional level. So what we start to see is there's not just a diversity in terms of different types of protists. There are different types of behaviors. There are different types of cellular structures. There are different ways of interacting with each other. Some of them can be solitary cells, unicellular. Some of them are cooperative as colonies, and some of them are fully multicellular with specialization within the body, within that multicellular body. And out of this, we're supposed to create a monophyletic clade. It's just not possible. It's just simply not possible. And so we have to come up with an alternative phylogeny. What has created much of this diversity is the role of endosymbiosis. So we've already discussed endosymbiosis once before. You've seen it in terms of the origin of mitochondria and the origin of chloroplasts as organelles within eukaryotic cells. Mitochondria get captured first through an endosymbiotic event, and as initially a beta proteobacterium that carries out cell respiration, and over time it evolves into the modern day mitochondria, in part by horizontal gene transfer from the bacterial precursor into the host cell's genome in the nucleus. And so the two cells now become dependent on each other, even though the mitochondria carries out a semi-autonomous or semi-independent behavior, producing its own proteins, having its own DNA, its own ribosomes in order to do that, dividing separately from the host cell by a bacterial form of division, binary fission, just divide in two. They are still dependent on each other. Neither can live separately from each other. So there's the symbiosis. Chloroplasts come along second as a endosymbiosis of a cyanobacterium. And that would actually give rise to two separate lines of protists that are photosynthetic, the red algae and the green algae. It's what happens after that that really creates a lot of, I don't like to use the term confusion because we're not really that confused in terms of what we think happened. It creates for I'll use the term complexity. It creates a lot of complexity in the phylogenetic tree, and here's the reason why. So here's the original endosymbiosis, which we now refer to as a primary endosymbiosis, the first endosymbiosis, and you'll see why in a moment. Out of that, this common ancestor as the product of endosymbiosis, you find the emergence of two lines of photosynthetic algae, the red algae and the green algae. Red algae evolve a red pigment as an accessory pigment to the chlorophylls. Why this then becomes a source of complexity is because we believe at some point following the emergence of these two lines of algae, a second symbiosis event occurs, where a eukaryotic cell symbiosis captures a green algae, and in another event captures a second line of eukaryotic cells captures a red algae. And the red algae and the green algae that are captured by a secondary endosymbiosis then give rise to a range of both photosynthetic and perhaps non-photosynthetic descendants. And there'll be more to come on that a little bit later in the presentation. But we can trace the plastids, the chloroplasts of one line or several lines of photosynthetic protists to a red algae. Their chloroplasts are clearly derived from red algae. So that tells us that there has been a secondary endosymbiosis of the red algae, with the accompanying loss of the vast majority of the cytoplasm, the nucleus, and all the other cellular material of that red algae. And the only thing that gets retained is the chloroplast. Everything else is discarded. In the second secondary endosymbiosis, the green algae is captured. Same thing happens. There's a loss of everything from the green algae. All the remnants of the green algae are discarded except for the chloroplast. That is retained. And so you have a second line of protists who have chloroplasts that are clearly derived from a green algae. What supports this whole idea of a secondary endosymbiosis is that there is one line of photosynthetic amoeboid cells. They belong to a group called the cercozoans, and they're somewhat unique in that they have a photosynthetic core inside the cell. And within that photosynthetic core, there is a remnant of a nucleus, along with a trace of cytoplasm, and of course, the chloroplast of the endosymbiont, the algae that was captured. what this suggests is that this particular endosymbiosis took place substantially later than the endosymbiosis, at least in evolutionary time, than the endosymbiosis that would give rise to the other line of green algae-derived protists. And I lost my train of thought for a moment. Let me just collect my thoughts again. And what is further interesting is that the group of algae, or the group of these amoeboid cells that contain this remnant of the algae, are capable of photosynthesis, and they do utilize the chloroplast for photosynthesis, but they also are heterotrophic. They do capture microorganisms. So they're behaving as an amoeba, but they're also behaving as a photosynthetic algae. So they're sort of in between autotrophism and heterotrophism. So they are actually referred to as mixotrophic. So this is another line of mixotrophs. But there is one member of this group that is strictly photosynthetic, even though it does have the remnant of the nucleus. It has all the other features of its relatives. It is strictly photosynthetic. It is not heterotrophic anymore. It's strictly autotrophic. So that again reinforces the idea that this is a unique secondary symbiosis that is separate from this secondary symbiosis line, and also separate from the primary endosymbiosis. And there's even a suggestion that, in fact, that this member of the chloroarachnophytes that is strictly photosynthetic may actually be the product of a primary endosymbiosis, because there is evidence of peptidoglycan around the remnant of the endosymbiont, which suggests, in fact, that the endosymbiont may have actually been a cyanobacterium and not an algae, a green algae. So this may actually be a second primary endosymbiosis, not a secondary endosymbiosis. So if you're starting to get the idea that the evolutionary history of protists is somewhat complicated, good, because that's what it is. It is very, very complicated. So what does the modern phylogeny actually look like? Well, here is our best hypothesis of eukaryotes, which includes the what used to be the protists. So anything that's highlighted in a yellow colored box is something that is derived from the Old Kingdom Protista. And you can see that there's a large variety of organisms here that were derived from the Old Kingdom Protista. But you notice also that the three truly multicellular eukaryotic kingdoms are still here, because they are part of the phylogeny of eukaryotes, so they have to be included, and they are included within the supergroups that they belong to. So here are the land plants. They're part of Archaeplastida because they are derived from one line of green algae known as the charophytes or charophycians. The fungi and animals are part of Uniconta, and specifically part of Epistheconta. Fungi are closely related to a line of protists called nuclearids, and we'll discuss those when we do the fungi. And animals are closely related to the choanoflagellates, and we will discuss the choanoflagellates when we talk about the animal kingdom, and I give you an introduction to the animal kingdom when we do chapter 32 towards the end of the semester. But everything else in the yellow boxes, apart from those two, we will briefly touch upon today. Now, one thing to notice, you notice that three of the supergroups have proper Latinized scientific names: excavator, Archaeplastida, Uniconta. We have one clade that's called the SAR clade, or the SAR clade. in quotes. And the reason why is because this clade has been formed by merging together two previously existing clades called the chromalveolata and the rhizaria. Now, within this clade, you still have the rhizaria as a separate group, but we've broken out the members of chromalveolata into two groups, alveolates and stramentopeals. They used to be recognized as the components of chromalveolata, but a more thorough genetic analysis in the last 5 to 10 years with the advent of gene sequencing machines has indicated that, in fact, these three clades do belong in the same supergroup, and it's fairly strongly supported at this point. But no one has come up with a formal name for the group. So until someone comes up with a more formal name for the group, it continues to be called the SAR clade. I often say that no one has really decided on a cool and sexy name for this group yet. However you want to look at it. So we still call this large group the SAR clade until someone comes up with a better name for it. So let's take our first break. Here are the concept check questions to surround this break. So remember, I will stop the recording here and join you on the other side of the break with the answers to these two questions. And I've got a typo here. What I wanted you to do here is to list at least 4 examples, or list 4 examples of structural and functional diversity found in protists. And then briefly describe the role endosymbiosis has played in eukaryotic evolution. So I'll stop here and I'll see you on the other side of the break with the answers to these 2 questions.